podcast, Pressurized, a short, punchy version of our main feed that gets right to the scientific point. If you like what you hear and you'd like to hear the full episode, you can find it in the same feed. And now, to get right to the point. I'm lucky enough to be joined by Thomas Desvignes, a fish biologist interested in fish living or adapting to extreme or changing environmental conditions. And he recently started working as an assistant professor in the Department of Biology at the University of Alabama at Birmingham. We got in touch because you were particularly interested in a disease that's been affecting the notothenoids down here in Antarctica. And you're very knowledgeable of the Antarctic fish. And I've been glad to see you appear in the dive stream a few times to help me with IDs.
I'm interested in really that fish disease, that's for sure. But originally, it's really more about the fish themselves. It's just that we stumble across that population of fish that was really infected by that new parasite. When we saw that, we just realized immediately that we had to do something. We had to sample and try to understand what was happening to those fish, because maybe about half of the fish we were collecting that night were having those sort of gruesome skin tumors. And turned out it was a new parasite, a new disease.
And it's a weird one, isn't it? It's an X parasite?
Yeah, XL. It's a unicellular eukaryote that just proliferates either in the dermis of the fish or in the gills. And it's actually known worldwide. The first fish that have been found with this type of XL disease were actually found in Norway and in Iceland.
That's how me and you originally made contact, because you were after any infected specimens to try and figure out what this disease was and how far it was spread. And the museum gets a lot of Antarctic specimens from the Ross Sea dependency as part of the fisheries observers. So we've got a little wanted sign up now, and we're going to keep an eye out for this strange disease.
The main idea is that when we started describing that species of parasites, we just realized that pretty much nothing is known about disease in Antarctic fish. Whereas we know a lot about diseases in penguins, in marine mammals, and other Antarctic birds, but fish, we don't really know anything. And the big thing is that fish are still really extremely important in the ecosystem in Antarctica. And so maybe with changing conditions, because Antarctica is changing quite fast nowadays, maybe those parasites or viruses may become more virulent, more prevalent in the environment. At the same time, maybe the fish are going to become a little bit more sensitive. And so all that combined, maybe it's going to be a big thing for the fish and the ecosystem. So because of that lack of information, we started to try to mention it to everybody we knew that was going to Antarctica and be like, hey, if you see these fish, just please make a sample. We'll analyze them later. We just need to understand better what's going on.
Alan was recently out here doing some of the deeper sites, and he commented that about 4,000 meters, loads of the Coryphaenoides rat tails seem to be particularly parasite-laden. We see hot spots of infection. I'm not sure whether it's population density. We noticed a lot more disease here, just in the deeper abyssal fish as well. I've been learning very quickly while I've been out here, seeing lots of really nice new groups of fish, actually. I've only seen them in the collection, so it's lovely to see things like the ice fish alive and swimming around and showing off their beautiful fins. I wondered if you could just talk us through the major groups that we're seeing here in Antarctica, because it seems they've all got quite a limited origin, and then they've radiated out into quite a lot of species here. So can you just talk us through some of the characters?
Well, the very main one are the notothenoids. That's really they dominate in number of species and also in number of specimens and biomass. The other big group is going to be the liparids, so the snail fishes, and the zooarcids, mostly the eelpouts. Those three groups are part of the perciforms, so they are somewhat closely related to one another. That's kind of interesting. And we find also a few skates and, like you were saying, some rat tail fish as well. We usually separate them in 5 families. Notonidae. Sorry, that's a hard one to pronounce because there's 2 I's in the middle. It's easier to write than to say. But those are the, for example, Traumatomys codi or Notothenia cordyceps, which are pretty common. Those are the red-blooded fish, but it's a family that is not monophyletic. So it's kind of debated whether we should maybe
separate that a bit more.
Yeah.
And they're kind of a classic-y looking fish. They've got scales. They tend to hang around at the bottom, or at least the ones I'm seeing are. They look a bit like the triple fins we get in New Zealand. They're a fairly fishy looking fish.
They kind of look like a classic fish in a way. A classic bottom fish. You know.
Yeah.
Like,
and then the other groups are a little bit more derived. They're a little bit more specialized. So can we talk through those?
yes. So then like a bit more derived, there are two groups of what we call the plunderfish. So the spiny plunderfish, which you probably won't see because those in Antarctica are represented mostly by one species. It's Harpagifer antarcticus, and it lives just subsurface in even tide pools. So you're not going to see that with Sebastian. The other group of plunderfish are the barbel plunderfish. And I know you've seen some of those, especially like Doloidraco longidorsalis. And those are really funny because they have this little chin barbel that goes out. Maybe function as a lure. We don't really know, actually. But they are funny looking fish.
They're definitely a sort of bottom living predator. So they look a little bit like a stargazer or a monkfish, especially with that lure, if that is what it is, coming out of the bottom lip. It did look very lury to me when I saw one earlier today.
Yeah, it is. And actually, I done a few histological analysis on those, and it's like jam-packed with nerves in there.
Oh.
Maybe it's a lure. Maybe it's some sort of like sense the environment. It's still really a mystery what it's for.
And there's quite a variation in them. Some of them are quite ragged looking and quite fleshy, and some of them are a whisker with a bulb on the end.
Yes, and even within one species, there might be some differences, some very tapered and some like almost looking like a cauliflower. So that's three. And then we get to another group that we call the dragonfish. So the Bathydraconidae. And that is subdivided in three subfamilies. And those are really interesting fish species as well. They're usually pretty small, maybe 20 centimeters, 25 centimeters long. They can live from close to the surface to like really deep, especially, for example, the genus Bathydraco has been captured like all the way down to like more than 2,000 meters deep.
Oh, and I think we've seen some here. Well, we almost caught one once, but it got out of the buy box. I was quite excited about having a Bathydraco.
Yeah, I think it might have been like Bathydraco antarcticus. There are 17 species of those. And I know that number pretty well because actually we just recently described the 17 species of them. The only character that differentiate them is the fact that they have only one dorsal fin compared to all other Nothothenioids having two dorsal fins. Some people are still debating whether it's a monophyletic family or not, because depending on the data set that is used to retrace the phylogeny, sometimes those three families don't form a monophyletic group. So there's still a bit of debate on that. But one thing that we know for sure is that they form the closest group to the ice fishes. And the ice fishes are probably some of the most odd fish that I would think of in the world, because they are actually the only vertebrates that we know of that live without hemoglobin. So their blood is transparent white. It's kind of like, I usually describe that as if you would put a few drops of milk into water and you stir that. And that's kind of like the color of their blood.
I've preserved a few that have come on board, and yeah, the pale white gills don't have scales. So there's like a semi-translucent feel to the flesh. They've got markings, but you feel like you can almost look through the fish.
So beautiful fish, though.
Yeah, they've really been winning people over. So if their blood lacks the pigment, like what's the makeup of their blood like? What cells do they have present in their blood?
So they have different types of white blood cells, and then they have thrombocytes. Thrombocytes is actually the equivalent of platelets in fish. Then they actually also have some red blood cells, like erythrocytes, but they are immature and they are in very, very small quantities.
So a bit of a evolutionary leftover. They still produce some seemingly non-functional blood cells.
Exactly. The genetic program to create those cells is not completely gone. Some blood cells that are like those stem cells from the blood, some of them still continue to differentiate into what we call pro-erythroblast. Just past that stage, they can like degenerate. But once in a while, we find some cells that are just immature red blood cells. But of course, they don't have hemoglobin because the genes are gone from their genome, like just gone. There's just one fragment of one gene left. It's been lost at one occasion in their ancestor. So, and that probably happened about like 5 to 6 million years ago when they diverged from the bathydraconids, from the dragonfish.
And they don't seem to be particularly hindered by it. These are very quick fish. They don't seem like they're running low on oxygen.
They are able to like really just like swim fast and escape if needed. And they're surprisingly more active than what we would think of.
Do we think this is an adaptation to the cold temperatures? It's strange they're the only group doing it. There's other fish successfully living here alongside them. So there must be an advantage to what they've decided to do here.
Well, that is a $1 million question. We are trying to understand what's happening and what happened and why this strange phenotype was preserved and spread into this radiation. Whether it's an adaptation, it's really hard to tell, because if it was really an adaptation, maybe other groups of fish would have done the same in Antarctica. So what we think mostly about this is that it's a disadaptation. It's something maybe that happened, a mutation, and maybe hemoglobin was not totally necessary because Antarctic water is really cold. And so there's a lot of oxygen that is dissolved. And maybe just by dissolution of oxygen in their blood, they were able to get enough oxygen.
A chance mutation that would usually be fatal, it didn't incur a fitness cost here. So they continued. And maybe there's even a bit of an energy saving as well. You don't spend so much energy producing hemoglobin, and maybe a bit of camouflage advantage as well, because there certainly is clearer for it. They don't have sort of strong coloration to them.
That is true. Maybe a bit of everything.
Yeah. Because evolution doesn't really have a plan. Just stuff happens. And then if it was a good idea, it hangs around. Now they've set up camp. They're doing really well. Within those, there's two main groups we're finding, at least, is the stilt-like ones, the ones with the really long pelvic fins that we've affectionately named the bipod fish here, because it looks like a tripod fish, but with only two. And then there's ones with more rounded pelvic fins that seem to sit a lot lower on the seabed. So the
bipod fish, it's actually the long-fingered ice fish. It's Cryodraco antarcticus that was actually recently renamed Pagetodes antarcticus. And those are really interesting. And actually, these fins, they have an allometric growth. Younger specimens going to have very, very extended pelvic fins, whereas the large adults going to have much shorter one compared to the body.
That's interesting. Okay, they catch up with them. Because I did see a lot of variation, but I didn't notice that, it's size related. Maybe a bit of a niche shift there. Maybe they are living like tripod fish and up off the bottom so they can eat invertebrates. And then once they put more size on, they can be more of a predator and be a sit and wait predator further down.
The other ones, yes, they sit at the bottom more. But a lot of ice fish as well swim into the water column during the day to try to find food.
Yeah, they do the vertical migration as well.
Actually, when I go to Antarctica, way to catch fish is mostly to trawl. So we use very small trawls, and we trawl for like very short amount of time, because what we want is to have fish that are alive and healthy. So we do really selective methods, but we always fish at night, because if we fish during the day, most of the time the net comes back empty because the fish are up in the water column.
How are they timing that? Because at the moment we don't have any night. It's been really disorientating here that it's never dark. I'm on night shift, but it's been lovely and sunny.
Yeah, it's very, very strange. And some researchers have started to study a bit of like the genes in the circadian rhythm. And they are finding that some genes seem to be missing in some species or might have evolved faster. It's very puzzling how they are able to sense the time of the day. Clearly, the seasons, because they go from it's light all day long to it's dark all day long.
And this is also the family that make the nests, we realized a couple of years back.
Yes, ice fishes, several species do nest. I think at least 6 or 7 species of ice fishes have been described nesting. A few species, though, don't. Like, for example, the mackerel ice fish, Champsocephalus gunneri, this species doesn't nest. We know that their eggs are buoyant and are released in the water column. But not only ice fishes do nest as well in Antarctica. quite a few species of notothenoids in general do nest. So dragonfishes, for example, several species have been shown to lay their eggs on stones, and the parents stay around and ventilate and protect the eggs. Clownfish as well.
So there's a lot of parental care in Antarctica.
It is. It's quite surprising the way they protect their eggs and attend. And that is maybe because some of the species, they actually don't lay so many eggs. They are producing very big eggs with a lot of reserves. And so they stay around probably to make sure that they give the best chances to those eggs.
And that tends to come along with almost direct or quite quick development. So that would usually indicate that the best place for the juveniles is where their parents were, rather than going up into the plankton and finding whole new areas.
Yes, totally.
It feels like we very recently became aware of just how extensive the icefish nests were. It was only a few years back. I think it was the German research vessel Polar Stern, but we had Autumn Purser on as well, who was one of the authors on that paper that revealed just these huge, huge fields of bowl-shaped nests with these icefish sitting in the middle of them. And I think it was the males guarding the eggs after mating.
Yes, that was just absolutely mind-blowing. Like seeing those images that they recovered, it was just absolutely amazing. And yes, it seemed that it was the male, but they also found like many instances where two fish were present on the nest. So maybe both parents were there.
And then the sort of outliers that we touched upon earlier in the main Antarctic groups is the zooacids and the loparids, the eelpouts and the snailfish, which I think we're still figuring it out, but I think they originated in the northern hemisphere and then seemed to find their way down. And then both of them just seemed to do really, really well in Antarctica and just radiated into loads and loads of species, which then seemed to have a bit of a deep sea advantage. And so seemed to radiate out from Antarctica into the deep sea zones. So that seems to be their path, although it's a bit muddy. They're taxonomically and genetically very tricky as well. We're just trying to tease all that apart. This is an important place for them, these iconic deep sea groups.
Yes, absolutely. Eelpouts and snailfish, I think they both are thought to have originated from the North Pacific Ocean. At least we worked recently with colleague with Joanna Katie from UC Santa Cruz. We worked on the eelpouts, and that was our conclusion, was that the group most likely originated from that area north of the Pacific Ocean and then dispersed themselves around the globe, mostly by probably the deep sea, just by proxy. And in Antarctica, indeed, there's a lot of those species and the diversity. The thing is that they are rarely caught. So there's a lot of species, especially snailfishes, that have been described based on only one specimen.
Usually very badly damaged, very fragile fish.
Yeah, they're mostly like goo.
Yes, yeah, extracellular matrix, but yeah, the technical term would be goo. They certainly melt and fall apart very easily. I managed to get my first one today with the slurp gun, so I'm excited for that one. So hopefully that'll be in quite good condition.
Yeah, I personally caught a few in one area on the peninsula. We are still trying to figure out names for them. One of them we found, it was Paraliparis megancus, and we are actually generating some DNA sequences for the species because it's almost absent in every single database. So that's also one thing that is very complicated, is that most of those species that have been described on like one or few specimens, they were preserved in formal, and formal prevents future DNA analysis. We know the species, it's very hard to key, but we don't have also like the genetic reference, which can be really helpful to actually see whether out of all those species, maybe some are the same species. It's just a lot maybe of intraspecific variability. So we don't really know yet. They're just hard to catch.
They're hard to catch. They're hard to ID. Depending on how you preserve it, even all the proportional measurements are different. They're so loose and gelatinous, and they lose all of that when you preserve them. Even two very similar looking fish, when they're captured, depending on how you preserve them and how careful you are, they can look very, very different in the museum collections and are very often damaged. So they're a nightmare group that I'm attempting to get my head around.
Actually, with some students, one year we raised some eggs from some snailfish eggs, because in one place when we were trawling, we got this, I think it was an hydrozoan, and there were eggs in that branch that had been laid there. I could tell it was fish eggs because I could see two black spots, like the
See
eyes
the eyes
of the little
Huh.
like embryo developing. We just kept them in our system for like 4 or 5 months. And after 4 or 5 months, they were still not hatching. Are they just like super slow developers and probably like takes a year or more for them to hatch? When they hatch, they are actually already looking like miniature adults.
It's direct development.
Exactly.
Did they ever hatch?
We were not able to keep them long enough, but we sampled them every 2 weeks. And actually, I'm working with a student right now to describe the development of these eggs.
Oh, that's really useful. And even that observation of how slowly they develop, that's going to be useful.
I hope so.
We've talked maybe a bit about some other adaptations we see. The obvious thing that these fish are up against is the cold. We've been seeing temperatures on these dives of 0.5 degree, 1.5 degrees, but the fish are surprisingly active. They don't seem sluggish, like you'd expect a cold-blooded animal to be behaving in such cold water.
We would just be like completely like dormant by those temperatures ourselves. The main thing for them to survive down in Antarctica is the fact that they have those antifreeze glycoproteins. That is an innovation of just that specific group of Antarctic notothenoids. It came from a gene, a trypsino gene, so produced by the pancreas, and it just evolved from that gene and became a repetitive structure that actually does not prevent ice from forming, but it kind of like comes and bind to the sharp edges of the ice crystal, preventing those sharp edges from growing and bursting open the cell.
Wow, I didn't know that. They're like smooth caps for the ice crystals to stop the sharp.
Exactly. Instead of being a very like sharp ice crystal, then it just makes them like be more round. But if you look in the cell of Antarctic notothenoids, they have ice in them.
That's very cool.
And so for that, you can read like all the beautiful work that Chris Chang from the University of Illinois in Urbana-Champaign has done. Her and her husband, Art DeVries, they are the one who described those antifreeze proteins in Antarctic fish. And they really understood well like the origin of the gene and how it works to protect the fish from freezing.
I can put it in the show notes so that anyone who wants to read that can.
Yeah, and what is really interesting is that Antarctic notothenoids are not the only fish that have antifreeze proteins, but all the other fish that have antifreeze protein, those antifreeze are different. Like for example, the cods in the Arctic, they also have antifreeze protein that allow them to survive in the frozen Arctic Ocean. But it's not the same gene. Eel pouts, some of them have also antifreeze. And again, it's a different one. So all those are like convergent evolution. Different ways evolution happen to converge into the same function, but by different routes. And even in cod, what Christina Chang has shown is that in cod, the antifreeze gene just came out from junk DNA. It was not even a gene.
That's incredible.
Yeah, like she described it, it's like it made sense out of nonsense.
What sort of temperatures can they cope with? How low can they go?
Like pretty much like all the way to the temperature of like freezing of seawater. And they can even like in some cases be what they call super cooled. They can be like super cooled to even like below the temperature of freezing of seawater, which is around like minus 1.8 degrees. So technically they can go below those temperature. But as soon as ice start to form at those temperatures, they almost like instantly turn into
the like snap freeze.
Exactly.
Yeah, ice builds on other ice far more efficiently. So as soon as you get that little nucleus, which also makes sense that they're almost doing like an oyster with a pearl. Like the ice crystals are forming and then they're coating it to stop it being an irritant and to stop it progressing.
When you think about it in Antarctica, especially in wintertime, in summer it's often true as well, the surface is colder than the deep sea. That at the surface, when you get in wintertime and the sea starts to freeze and sea ice starts to build, it's because it reaches -2 Celsius, whereas you don't see those temperatures down deep. Even by, let's say, 600-700 meters, at the surface, we were like at -1.8, -2, and then going down And by 700 meters deep, we were maybe around like 1.5 degrees.
They don't tend to come down this far, but there are some commercial fisheries in Antarctica, and it's mainly long lines, I think. In fact, that might be the only type that's allowed.
If we are talking about like finfish fishing, yes. The only species that is really targeted is the Antarctic toothfish. It's very regulated where they are allowed to be fished. Actually, most of the fishery happens in the Ross Sea. There's very little that is done on the peninsula. There's quite a bit more around South Georgia.
I've not seen any lost gear or any filament out here. And to be honest, we've seen absolutely no litter, which is a hard thing to do these days. I can't remember the last time I had an expedition where we didn't see any litter.
It's not completely absent, but it's pretty clean environment still. Most of the fishery that is happening right now, at least on the peninsula, it's about krill. It's the krill fishery on the peninsula.
And that's quite new, isn't it? We didn't really exploit krill. To me, at least, it feels the last few years.
I think it's probably really boomed maybe the past decade, but now it's a really important economic activity on the peninsula.
And it tends to be supplements, isn't it? I see a lot of health food supplements based on krill.
Yes, it's for health supplement, but also for animal food.
That seems a strange place to go to get it, but I guess protein is protein.
Yeah. The other fishery that exists is actually an ice fish species like Champsocephalus gunneri, the mackerel ice fish that is also commercially fished around South Georgia and around the Kerguelen-Heard Plateau.
Before we finish up, I wanted to talk a little bit about your science communication, in particular the comics that you've been putting out.
Yeah, that has been really fun to do.
And I'm loving how many languages you put them out in. Science in general and science communication, unfortunately, is dominated by English. So it's really nice to see options in a variety of languages.
Yeah, well, that is all because of great collaborators around the world.
Well, we definitely link to those.
Yeah, you should. Thank you.
We will. Thank you so much for your time, Thom. It was really lovely to talk to you.
Oh, likewise.
Coffee's ready.
So we're not going to let the whole Pacific get in the way of a coffee with Andrew.
No, hell no. I'm gearing up to go to sea, so the marine team will be all at sea. 3 men in a boat, brackets, different ones.
Yeah, 3 trips. So Kerry, who's been on the show before, our malacologist, mollusk expert, he is currently out on the Sonne, the German vessel.
You're bouncing around the Antarctic Peninsula and Bellinghousen Sea, and I'm getting ready to go down to the Pusiga Trench.
Now, this is a rare one. This is one that doesn't get mentioned very often. So the same plate boundary that makes our beloved Kermadec Trench to the north has a sinister twin to the south that no one talks about.
The evil twin. The plate boundary comes down through the Kermadec and then runs basically down the Hikurangi Trough, and then does a sharp dogleg, and the fault line goes down the mid-alpine fault. And then it peels off the bottom southwest corner of New Zealand to become the Pusiga Trench, and then a little bit further south in the Australian EEZ, you have the Hiort Trench. To my knowledge, neither of these trenches have had any kind of biological investigation or specimens taken from them. No critters, all new.
You excited?
A mixture of excitement and apprehension. You know, you're stuck in this little sphere. I think the thing that I'm most apprehensive about is cramp. You haven't actually got a lot of space to stretch out a leg. I might just have to take a magnesium tablet or something. I've asked Daniel, he says, it's like sitting in a big armchair.
I do hope you get a shot.
Oh, yeah. Yeah, this is bucket list country here. But you got some snailfish. You've come across Snailfish Central.
We were right close to the ice shelf, and loads of notothenoids, loads of ice fish and things, the usual characters. And there was one sighting of just the back half of a snailfish sort of leaving the shot. But now we're out by the shelf break, seeing loads of them. They should be up in a few hours. They'll probably be alive. So I'll do some filming of them in the aquarium where the gel is nicely suspended. Snailfish, they're made of goo.
Yeah.
And they never preserve quite so well.
They can preserve really well if you put them fresh, dead into formalin. It seems to fix the gel. But if you freeze them, then that's just, you wind up with something that looks like an old sock.
I think the ice crystals just break it all up. Whatever sort of cohesion there is, I think the ice breaks it.
We had David Stein out here working on our book. He was telling me that there's one species in the Ross Sea that is only found in this one bay. He said, you go there, you put the camera down, and you just see them everywhere. And he wonders whether or not there's a bit of co-reliance on some of maybe the invertebrate fauna. Maybe they're relying on the sponge fauna that's there. The zooarchids do. Some of them have been filmed in the sponges. They even lay their eggs in the sponges, because if you've got a great big silicious sponge, no predator's going to stick his snout into what is essentially a cactus to try and get a few eggs.
We've been sticking our snoot in the odd large glass sponge when we've been on the ROV, because you'll quite often peer in with the camera and there's a load of shrimp looking out at you.
Yeah. Interesting, you've been getting a lot of barracudinas.
A lot. I haven't asked for them. They sample themselves.
Yeah, that's brilliant.
They wedge themselves in the ROV.
They're just stunned. And I've been impressed with how fat some of them are. I don't know whether they're full of food or full of eggs or both.
Yeah, we'll have a look at them maybe when I bring them up.
So when do you actually finish work on the Falkor 2?
So this is the final site that we're at the moment.
Mm-hmm.
Then it's a fair trek back to port.
Mm-hmm.
It's a week just to get back. So we'll grin and bear the Drake Passage. We were fairly lucky on the way down.
Mm-hmm.
And then a little bit of a demob. And then I've talked my way onto one of the tourist expedition ships.
Mm-hmm.
So then going out for another 2 weeks out of Ushuaia to continue hunting squid. And Kat's going to join me for that.
You've obviously enjoyed yourself far too much.
It's been hard work, but it's beautiful. I see why people fall in love with it down here.
Yes, that's what struck me. You get down there within an hour, you're going, I get it.
I get it. Yep.
No picture and no video actually captures just how incredible that place is. You just felt so privileged to be there, to be able to be part of this and to make these incredible discoveries. Like I say, I would go there again in a heartbeat if I had the opportunity.
So until the next episode, we'll deep see you next time, and I have this you already.